Hello everyone, welcome to our YouTube channel. Today we'll be covering receptors of general sensation. And remember the receptors that we're going to cover today are actually our sensory receptors of general sensation. For the next video, we're going to cover receptors of spatial sensation. So welcome. For the scope of the session today, we are going to define various terms, including what is a receptor, what is a sensation, and what is a sensory organ. And then we are going to understand the classification of various receptors. And then we're going to describe the receptors of general sensation. And we look at histological images of each. So for this slide, what you are being able to see are you can appreciate the various receptors. So starting with the Merkel disc, then you have the Ruffini endings. Then you have the Pacinian corpuscles. You have the free nerve endings, the Krauss end bulbs, and the Meissners. So skin is one of the sensory organs. You can appreciate the skin with epidermis, enterodermis, but we'll look more into this in a few moments. So if you're going to define various terms, we'll start the first thing with defining what is a sensation. So sensation is that state of perception, the state of awareness of a particular stimulus. So when you perceive the coldness, when you perceive the hotness, when you perceive the pressure being applied on your skin, that's a sensation. And then that sensation, that stimulus can either be external or internal. So for example, the light that comes from the sun is external. But when you have extension or distension of the stomach, that's something that is internal. So the stimulus coming from the body, within the body. Types of sensation we have two, can either have somatic or spatial sensations. So somatic are sensations that comes from all over the body, and they are relayed by spinal and cranial nerves, while spatial sensations are sensations that at this point come from head and neck. They come from particular organs. And in addition to that, they are relayed by cranial nerves. So light sensation, auditory sensations, olfactory sensation, and smell sensations, they are actually perceived by these special organs found in particular organs, and then they are relayed by cranial nerves. Even for somatic sensations, they are relayed by spinal and cranial nerves. So we have special sensations and somatic sensations are the only two types of sensations. For this image, I'd like us to appreciate on how a sensation at this point is perceived by a sensory receptor. Then from there, it is taken by the various tracts into the higher center. So at this point, you can appreciate this is a free nerve ending, perceiving pain. Then pain goes through first order neuron, and then it decassetes in the spinal cord, then it ascends through the spinal thalamic tract. Then it releases the second order neuron ends in the thalamus, and then you have now the that order neuron going in the somatosensory cerebral cortex. So at this point, you can appreciate that that is a pathway. So you can have pain sensation, you can have vibrations pathway, you can have pains pathway, you can have so many pathways regarding which modalities you have been carried by specific tracts, specific pathways that we described a few days ago. So we're going to classify various sensations, and the sensations can either be mechanoreceptive. So these sensations are actually coming due to mechanical displacement of the body. So such sensations include touch, pressure, and positional changes. So those are mechanoreceptive types of sensations. Sensations that comes due to mechanical displacement of the body. So in the skin, you can have tactile displacement, you can have touch, you can have exertion of pressure, you can have positional change of the body. That's mechanoreception. That's a mechanoreceptive type of sensation. Then you have the thermoreceptive sensations. These are sensations that are coming due to heat or changes in temperature. So when you're moving from a low temperature to a region of high temperatures, that's a thermoreceptive sensation. And the last sensation is pain sensation. And then this pain sensation ideally comes from due to the damaging effects to the body tissues. So when you have excessive heat, that's a pain sensation. When you have now an acid damaging the body, that's a pain sensation at this point. And the pain sensation at this point is a third type of sensation. So remember the three types of sensations: mechanoreceptive sensation, thermoreceptive sensations, and the pain sensations. There are three sensations. Now we're going to discuss receptors. So we've described what is a sensation. So that state of awareness, that state of perception of a certain stimulus is what is known as a sensation. Then you've gone ahead and described that stimuli can either be two, external or internal. Then we've went ahead and described the various types of sensations: mechanoreceptive, thermoreceptive, and nociceptive at this point. What is a receptor? So a receptor is any structure that can be able to detect a stimulus. So there is that point where this receptor, for it to be called a receptor, must be able to detect a stimulus. And it has a specific characteristic. It needs to be a transducer. So when you talk about a structure being a transducer, it has to convert environmental stimulus to bioelectric stimulus. So this bioelectric stimulus is the action potential. So transduction is changing of one form of energy to another, and then this receptor is changing environmental stimulus to bioelectric stimulus. That's the reason why it is a transducer, as receptor is a transducer. So you can classify receptors in three, considering three factors in terms of the structure. How does it look like? A location, where is it located, or where is the stimuli coming from, or the stimuli, where does it come from, and the location of the receptor. So in terms of the structure, we have three classification. You can either have free nerve endings, which at this point they are unmyelinated. You can have the free nerve endings being encapsulated, giving us the second type of receptor in terms of the structure, the encapsulated nerve endings. And then you can have now specialized receptor cells. So these ones, they just have a free nerve ending going to a specific cell. So the three types of receptors in terms of structure, we have free nerve endings, which are unmyelinated, encapsulated nerve endings. So that means the free nerve ending that is unmyelinated is covered by a capsule. And then you have specialized receptor cells. That is, you have a free nerve ending going to a specific receptor cell. So, according to structures, according to the structure of the receptor, we have three types of receptors. According to stimuli, we have a couple of them. We have five. So mechanoreceptor, this one at this point, the stimuli are mechanical forces. So touch and other things are mechanoreceptors. Then you have other receptors which are thermoreceptors. So these ones detect changing body temperature, for example, coldness and hotness. So at this point, we have Krauss and bulb and Ruffini for heat. Then for nociceptors, this one, ideally they detect damage in tissues. So nociceptors always goes hand in hand with pain sensation. So pain is a stimulus at this point, and the receptors are nociceptors. And then photoreceptors are a good example is light now. The cones and rods found in the eye, the one that can be able to convert the electromagnetic stimulus at this point, photoreceptors of the eye, rods, and cones. And the last one is chemoreceptors. So these chemoreceptors at this point, they check the concentrations of various chemicals, blood sugar, taste, pH, smell, osmolality, uh, all those, they are actually chemoreceptors. They check the concentration of chemicals, the concentration of metabolites within the body. So through that, they can be able to enable the body to regulate maybe the blood pressure, regulate the blood sugar, regulate blood glucose, regulate the taste, smell, and other things. According to location, you have two extoreceptors and introreceptors. So extoreceptors, so that means the stimulus is coming from externally, external, outside the body. So that means the receptor, yes, is found on the body surface or next to the body surface, but the stimulus is coming from outside the body. So that means the receptor must be found within the location. So maybe at this time you can try to give some of the examples in the chat box. You can give examples of extoreceptors. Then you have interoreceptors, which are also known as visceral receptors. And these visceral receptors, that means the sensations are inside the body. So they are found in the body organs. That's the reason why the receptors are found also within the bodies. And they detect changes in blood pressure, changes in stretch, changes in pressure, temperature changes, and so forth. But ideally, the point of operation is that these interoreceptors are known as visceral receptors because they are found in various viscera, and viscera are organs at this point. What are then are these properties of receptors? So we have 5 properties of receptors. One of them is that there is excitability of the receptor. So the receptor must be able to generate an action potential. So the receptor potential must be converted to an action potential. That's one of the characteristics of receptor. The other receptor is that it has adequate stimulus. You must have an adequate stimulus for you to generate an action potential. You need to have a stimulus that can be able to reach a certain threshold and generate an action potential. That's the reason why adequate stimulus is a property of a receptor. Then specificity of receptors. For example, photoreceptors, they are for light. Olfactory receptors, they are for olfaction. So they have specificity. Then there is that intensity discrimination. So where we talk about that, for intensity discrimination, you need to have a certain number of receptors being stimulated for you to generate an action potential. You need to reach a certain threshold for you to generate an action potential. And then these action potentials, their frequency must be increased. There's a certain way that you can have action potentials, yes, but you can't generate a stimulus. That stimulus can't generate an action potential. So in terms of intensity discrimination, that receptor or those number of receptors or a certain number of receptors must be stimulated for it to generate an action potential. And then for those action potentials, they need to be generated frequently for you to reach a certain threshold for you to send a sensation to the brain that yes, your hand is in fire or something like that. So that is intensity discrimination. Then the last one is receptor adaptation. And for receptor adaptation, that is reduced responsiveness followed by prolonged action. A good example is when you wear maybe a shirt, at the first moment, you realize that yes, I've worn a shirt. But with time, due to prolonged action, due to prolonged wearing of that shirt, that awareness of wearing the shirt, ideally at this point, is reduced. So that is reduced responsiveness in that bit. That is receptor adaptation. So excitability, adequate stimulus, specificity, intensity discrimination, and receptor adaptations are the 5 properties of receptor. Then remember these receptors, are actually organs of transduction, as structures of transduction, as we noted earlier. Then the last definition that we'll have today is what is a sensory organ. And a sensory organ is any organ that houses a receptor. So, for example, the eye. The eye houses the retina. The retina contains rods and cones. So the eye is a sensory organ. Come to the skin. The skin houses Pacinian corpuscles, houses Meissner's, houses free nerve endings. So that's a sensory organ. Come to the ear. It houses the hair cells. It houses structures for equilibrium and structures for auditory sensations. So the ear is a sensory organ in that bit. And at this bit, we can give examples of sensory organs in the chat box or in the comment section. We'll go ahead and check whether we got them right. So we look at receptors, and some of the receptors that we cover today, tactile receptors, at this point you can approach it. Here you have a receptor being pointed. Approaching is a skin, this is a keratin, then you can approach the epidermis, this is a dermis. And then on this dermal papillae, we have this receptor here. This makes Nancy receptor. Then you have this very good image of Pacinian corpuscle. This is surrounded by fat. It has a capsule. Then you have concentric rings looking like an onion bulb. And then you have a free nerve ending that is unmyelinated at this point. So that is a Pacinian corpuscle. And then this is Meissner's corpuscle. You can see it's capsulated and the free nerve endings are pointed there. So free nerve endings are one of the tactile receptors. And at this point, they're found on the skin and non organs. They perceive touch in another type of sensation, such as pressure and touch and the rest. For Meissner's corpuscles, I showed us this is a Meissner's corpuscles. They are found in the dermal papillae. These are dermal papillae, and they perceive fine touch. That is very important. Fine touch for the purpose of exams. Fine touch is perceived by Meissner's corpuscles. Then the other one is Merkel disc. I will show us an image on the Merkel disc. So this one perceives the texture, perceives crude touch. Remember, Meissner's corpuscles was for fine touch. Merkel's disc is for crude touch. And then peritracheal nerve endings, the nerve endings that are found on the shaft of the hair. So they just detect, they perceive sensations that similar, which is movements of the hair. So they perceive movement of the hair, and then they tell you that yes, it's a cold environment, it's a warm environment. And then the other type of receptor is Ruffini. So Ruffini receptors, at this point, they are found in the reticular dermis. They give joint movements, they give joint locations, and then in addition to that, they can be able to perceive warm sensations. But the most important one, in terms of tactile sensation, is that it allows joint movement perception. And then we have Pacinian corpuscles. And Pacinian corpuscles, they have concentric rings, as you saw here, which looks like an onion. And then they're found in the subcutaneous tissue, as you can see here. So subcutaneous tissues of mesentery found in the skin, they're found in other structures that will tendons and muscles. So that is subcutaneous tissue, Pacinian corpuscles, which perceives vibration and pressure. So these Pacinian corpuscles. So Pacinian corpuscles for vibration and pressure, Ruffini for warm sensations, peritracheal for movements of the hair, Merkel disc for crude touch, Meissner's corpuscles for fine touch, and free nerve endings for pain, majorly pain, but also it perceives touch and pressure. Proprioceptors, we have 4 proprioceptors, but we'll dwell more on 2. One of them is here. So this is a neuromuscular spindle. You can see we have 2. The neuromuscular spindle is another neuromuscular spindle. So there are 2. We have these neuromuscular spindles. They are actually found on skeletal muscles. So these are skeletal muscles, the extrafusal muscles of extrafusal fibers of skeletal muscles, extrafusal fibers of skeletal muscles, and then you have neuromuscular spindle with a capsule encapsulating it. And then what is inside are actually intrafusal fibers. And we will see the fibers that are found in these, the types of intrafusal fibers found in the neuromuscular spindle. So one of them, muscle spindles of the neuromuscular spindles, which detect stretch of the muscle. Then you have the Golgi tendon organ, and this Golgi tendon organ, at this point, it perceives tension within the myotendinous junction. So remember, this is a muscle, this is a tendon, and then this junction is a myotendinous junction, and that is where you have the Golgi tendon organ, or the organ of Golgi, showing ramification of nerve fibrils. So those are actually the Golgi tendon organ. It detects the tension within the muscle or within the junction at this point. And then the other two receptors for proprioception, at this point, they don't give that joint position more. We have Pacinian corpuscles and Ruffini receptors. For nociceptors at this point, is that we have free nerve endings. And these free nerve endings are being shown on the image on our right. Is a free nerve ending going to the skin. So this is the skin, this is epidermis. You can see these are free nerve endings to perceive touch, pain, and pressure, and the rest of the receptor of the sensations, the rest of the stimulus. These are free nerve ending, which is unmyelinated. Free nerve endings. These nociceptors, the free nerve endings, um, are found in the skin, periosteum, meninges, joints, and any other organ in the body, but they are found in higher concentration in the skin, periosteum, meninges, and joints. That doesn't say that these free nerve endings are not found in any other location. They're found in all the body parts, but they're found in higher concentrations, in high numbers in the skin, periosteum, meninges, and the joints. And they can detect any damaging stimuli. So high temperatures, high pressures, a lot of vibration, all those cold temperatures are perceived by free nerve endings. And now the same points, uh, as we continue to share the content, can you like, comment, share, and subscribe as we try to look for more content. Then the other type of receptor are actually the thermoreceptors. So three of them, the three thermoreceptors, free nerve endings, Krauss and bulb, and Ruffini. So free nerve endings, at this point, they can be able to detect warm temperatures, low temperatures, that is cold temperatures, and pain receptor. So free nerve endings, they perceive three sensations or three stimuli. So cold temperatures, warm temperatures. Free nerve endings can also detect pain. But now at this point, because they are thermoreceptors, they can be able to detect coldness and the degree of warmness. So degree of coldness and the degree of warmness can be able to be detected by free nerve endings. So we have free nerve endings specifically for low temperatures, intermediate temperatures, as you rise towards warm temperatures and even heat temperature, like high temperatures at this point. Then receptors for cold temperatures, or they can be able to perceive the degree of coldness, actually cross and bounds. And then the receptors that can be able to perceive heat or degree of hotness is the Ruffini receptors. What you're seeing at this point, this is the Krauss end bulbs. This is the Krauss end bulb. Then the other one that you are seeing there at this point, this is the Ruffini receptors. the Ruffini receptors. And the Krauss and Barber, you can see, and the Ruffini, they're both of them. And they have receptors. As you can see, they have receptors. They have free nerve endings. So the free nerve endings, in addition to that, you can appreciate they're encapsulated. They're encapsulated. So in terms of the structure, you classify Krauss-Enberg and Ruffini as capsulated types of receptors. And then free nerve endings, they're just free nerve endings, which are unmyelinated at this point. Then we look at this. I presume you saw the location, we saw the Pacinian corpuscles found in the subcutaneous tissue. We have Ruffini corpuscles for heat, Krauss-Enberg for cold, Meissner's receptors for fine touch. We have peritracheal root hair plexuses, peritracheal stroke root hair plexuses. At this point, they detect the movements of the hair, and then you have free nerve endings to perceive coldness, hotness, and pain. Buccal discs for crude touch, and that is being shown on the image on our left, with the free nerve endings for the three stimuli, Buccal disc for touch, crude touch, those and bulbs for touch, and also for degree of coldness. Root hair plexuses for any movements that happens on the hair due to touch. And then we'll have Pacinian corpuscles for pressure and vibration, Meissner's corpuscles for fine touch, Ruffini endings for degree of hotness and also pressure. And the first one that you'll observe today is Pacinian corpuscles. So you can see Pacinian corpuscles found in the subcutaneous tissue. In addition to that, we can be able to observe that it's lamellated, it has concentric rings, then it has a capsule. So we have three Pacinian corpuscle in this image found in the subcutaneous tissue for vibration and pressure. Then the other one, the neuromuscular spindle. This is a neuromuscular spindle, all the muscle spindle. You can see it's found actually on the skeletal muscle, the skeletal muscles. You can see nuclei on the periphery of the fibers. So these are extrafusal fibers, these are capsule, and these are intrafusal fibers with free nerve endings in it. So remember, neuromuscular spindle, they detect stretch of the muscle. On the image on our right, you can be able to appreciate Meissner's receptor, Meissner's corpuscles, found on the dermal papillae. So this is a receptor. You can appreciate this epidermis, this epidermis, this is a receptor, it's a dermal papillae. And appreciate the Meissner's corpuscle is capsulated, and the free nerve endings are present, and it perceives fine touch. So Meissner's fine touch, Pacinian vibration and pressure, neuromuscular spindle for stretch. So at this point, maybe you would like us to tell us what is that being shown on that image. So what is this receptor here? So that receptor is actually free nerve endings. So I won't discuss much because we talked about it. There's a free nerve ending for all the structure of the body, but you can appreciate the skin. So that's a free nerve endings. And this is the Meissner's. You can appreciate this Meissner's corpuscles. You can appreciate it there. Same case is the Meissner's corpuscles. This is a tactile corpuscle or the Meissner's corpuscle being found on the dermal papilla. You can appreciate the structure of the skin on that slide. Yes. This one we'll discuss it more. Can appreciate. Remember when you're talking about classification of the of receptors in terms of the structure, you talked about a free nerve ending which is unmyelinated. We have capsulated free nerve endings. Then you have specialized receptor cell. And one of the specialized receptor cell is the Merkel cell from nucleus cells. So these are Merkel cell and an elongated or an enlarged nervous tissue, nervous fiber. So you can see it there. So the two, the Merkel cell and the nerve fiber, they make up the Merkel disc. And the Merkel disc at this point perceives crude touch. The same thing is being depicted there. You can appreciate this a Merkel cell on stratum basale, and there's a free nerve ending. So the two, they make up the Merkel disc. So Merkel cell is really found on stratum basale and the epidermis. And then this is actually a Ruffini. It's a Ruffini receptor for hotness, for warmth. So you can appreciate the ending, can see encapsulation there. Then all that make up a receptor, which is Ruffini. For the rest, this Basilian, you can appreciate, we've talked much about this Basilian corpuscle. That is direct. I presume at this point you can be able to recognize that is Basilian corpuscle. What are the parts of Basilian corpuscle? You can see it has the inside terminal end is non-myelinated. Then as you move towards now, the nerve fiber is myelinated also. So this at this point you're just perceiving the sensation. Then at this point you are conducting. That's the reason why we have myelination by the Schwann cells. Then inner laminations, outer laminations making up the concentric rings to make it look like it's an onion bulb. Then that's the capsule of the Pacinian corpuscle for vibration and pressure. Golgi tendon organ T, tendon T, tension. So Golgi tendon organ perceives tension within the tendon, the myotendinous junction. So you can see it here. So it's one of the proprioceptors. At this point, we can name in the comment section, we can name, first of all, what is this receptor here? And then what are the various parts of this receptor? Because you described it actually a few seconds ago. So what is this type of a receptor? And what are the parts of this receptor? So what is part one? What is part two? And what is part three? Maybe in the comment section, let me know. And we'll describe this, but as we describe it, this is a neuromuscular spindle. The neuromuscle spindle, the muscle spindle using H and D stem, and we have the extrafusal fibers, which contract. And then we'll have a capsule covering this. So a capsule together with the intrafusal fibers, they make up the muscle spindle. And then these intrafusal fibers, muscle fibers, at this point, you will be either be having a nuclear chain and a nuclear bag. So what you're seeing here are actually nerve fibers. Muscle spindle for stretch of the muscle. And that is being perceived here. You can see extrafusal fibers. This is a muscle spindle. You can appreciate the capsule. And then you have inside it, you have the intrafusal fibers. So extrafusal fibers are just a normal skeletal muscle which you contract and relax normally, because you can appreciate here the nucleus on the periphery, it's cylindrical in nature, and then they are multinucleated at this point. So this is just found, they are just in the skeletal muscles for stretch of the muscle. This is 2D. The first image is actually 2D. Then three-dimensional of the image is being shown there. So the reason I put this image here is that we can be able to appreciate the innervation in parts of the intrafusal fiber. So muscle fibers, which are actually outside, they are outside the muscle spindle. That's why they are known as extrafusal, external fibers. Muscle fibers, which contract and relax, are extrafusal fibers. Then we have fibers that are inside the muscle spindle. That's the reason why they're known as intrafusal fibers. And you have two types of intrafusal fibers. You can have nuclear bag fibers and nuclear chain fibers. So nuclear bag fibers, they are thickened, like the bag is thick, as you can see here. So that's the reason why they're known as nuclear bag fibers. They are thickened. And then nuclear chain fibers, you can appreciate they just look like a chain. It's like a straight line. a chain connecting one fiber to the rest. So nuclear chain fibers. So in terms of innervation, the extrafusal fibers and intrafusal fibers will be innervated differently. So as you can narrow down, you can appreciate is a myotendinous junction. This is a muscle, this is a tendon, this is a myotendinous junction. And actually you can see the Golgi tendon organ, the GTO, which perceives tension of the myotendinous junction. Then you have the perimysium, narrowing, covering the a fascicle of a muscle. Remember, now the neuromuscular spindle will be found inside the muscle. So muscle fibers that are found outside the capsule of the neuromuscular fibers are known as extrafusal fibers. Fibers that are found inside the neuromuscular spindle are known as intrafusal fibers. So extrafusal fibers will be innervated by alpha motor neuron, efferent alpha motor neurons. Then intrafusal fibers will be innervated by gamma motor neurons. So they'll be innervated by gamma motor neurons. And for the efferents now, efferents coming from neuromuscular spindle to the efferents coming from efferents coming, okay, at this point, efferents coming into the intrafusal fibers will be gamma efferent motor neurons. And now the afferents that will be coming from the neuromuscular spindle will be annulospiral and flower spray types of neurons. So annulospiral and flower spray will be coming from this fiber. So you can appreciate flower spray is actually this flower spray type 2 sensory endings. And then we have now the annulospiral. They'll be winding along the intrafusal fibers. So annulospiral type 1A sensory endings. So type 2 sensory endings are allow flower spray. So that is the innervation in terms of extrafusal fibers, intrafusal fibers as afferents and efferents. So kindly be aware of that. Maybe to wrap it up, can appreciate is a Krauss end bulb, capsulated also for cold, this Krauss end bulb. We can appreciate this Ruffini, it's encapsulated. Maybe at our own time, we can be able to name what is one, a free nerve ending, what is two, it's the name of that fluid that is found there, and then what is three, the capsule, the name of the capsule. So name the three parts of Ruffini. Maybe in the comment section, let me know the parts of Ruffini for hits. Then can appreciate this is a very beautiful image of Pacinian corpuscles, a magnified image of Pacinian corpuscles. Can appreciate is a free nerve ending at the center, various lamellations, the concentric rings, and then you can appreciate the capsule. So Pacinian corpuscles for pressure and vibration. And then we also saw this is a neuromuscular spindle in this point. Yeah, so that is it. According to Colossians 4:23-24, like whatever you do it wholeheartedly. Do it as if you're not serving earthly masters, so that at this point, whatever you do it wholeheartedly, so that you know the blessings and the inheritance will come from the Lord Himself. So that is it. Thank you very much for the session. See you next time. Kindly like, share, comment, subscribe, and even tell your friends and colleagues about it, about the YouTube channel. We'll be proud to grow together. So thank you for your time. All the best in your endeavors.